much, Marika, for this very nice introduction. And thank you, everyone, for inviting me and giving me a chance to present some of our work. I hope that everyone can hear me well and also see me, and then hopefully my slides very well. So let me try to share the slides. I'm assuming that this works, and otherwise you have to stop me. Okay, so we are interested in tumor evolution, and in particular in modeling this process from single-cell sequencing data. So we have a very evolutionary perspective on oncology and on interpreting omics data from tumors. And so the way we think of this process is simply that there are alterations occurring in individual cells that give these cells initially a proliferative advantage. And while these cells keep replicating, they can accumulate additional mutations indicated here by the different colors. And this process can and may eventually lead to the formation of a carcinoma. So tumor cells may become invasive, leave their tissue of origin, and then potentially even form metastases in other parts of the body. And at the carcinoma stage, typically this process becomes clinically relevant. We're able to detect these tumors, and of course they cause all sorts of problems. Now, the same process is illustrated here, where the normal tissue is on the left and the first mutation occurs here, and the different clones that evolve now over time are represented in different colors, and their size is indicated. Such a clone may also go extinct again. Others will eventually dominate the population. And now if you look at such a tumor at a certain point in time, so you sample the tumor, you obtain a biopsy, and you can look at the genetic composition of that tumor, that allows you basically to make inference about the evolutionary process that has given rise to the tumor that we observe. So we would like to reconstruct from a single time point, typically, the evolutionary history of the tumor, and to assess the diversity that we observe within the tumor. Now, the way tumors evolves is very heterogeneous, and it depends on a lot of factors. Among others, it depends on the spatial structure for solid tumors that these tumors have, and of course the tissues they originate in. It also depends to a large extent on mutational processes and on the underlying fitness landscape, so on the selective advantages that different mutations or different combinations of mutations confer to such a tumor. And depending on what this mode of tumor evolution is, we will see different patterns in the tumor phylogeny when we try to reconstruct the history of a tumor. As indicated in the last column here, you see very different phylogenetic trees, where also the clone size is represented by the size of each node. And just to get a sense that these patterns can be very different and can be very diverse for different types of tumors. Now, before going into how we do this, I want to make a few comments why this question is also clinically almost immediately relevant. So starting on the left-hand side here, this is an early paper from Carlo Meli showing that tumors that have a higher genetic diversity, which is here quantified as the number of different clones that have been identified within the tumor, then actually they have a poorer prognosis, so they have a higher chance of progressing to the carcinoma state, as opposed to patients that have a lower genetic diversity. And not so long ago, but also a few years ago, there were the first papers analyzing the order in which mutations occur. So here's a particular example of mutations in two genes, TAT2 and JAK2. And again, it turns out that if you have TAT2 first, then progression of the disease is much more likely, so the prognosis is poorer. All of these patients have the same combination of these two mutations, but just the order in which they occurred differs between the two patients. So that means just the amount of diversity on the left here, irrespective of what the mutations are, matters for clinical outcomes, and also the order of the same mutations matters for clinical outcomes. So we would like to assess these properties from genomic sequencing data. Now, I've been very lucky to be part of a project that I want to mention briefly here, the so-called tumor profiler study. This is a large effort in Switzerland involving many research groups from hospitals, from basic research institutions, also from industry, where tumors are analyzed with a whole range of different technologies. And these data are produced in different labs. The data is then integrated and analyzed in a joint fashion, such that with a total turnaround of 2 weeks, we obtain results from these different technologies, and no later than 4 weeks, these results in the form of condensed reports will also enter the molecular tumor board and eventually attribute to clinical decision making. Now, in this project, one of the technologies refers to single-cell DNA and single-cell RNA sequencing. So that is the technology we have been involved in and we have been working on. And I want to show you some of the things that we have done in the course of this project. So let me go back now to the tumor phylogeny problem. So again, we're starting with the tumor, which consists of different types of cells that are genetically different, and I will only focus here on the tumor cells. So they carry different combinations of mutations. We are taking a sample at a single point in time, and we will only care about single cell sequencing data here in my presentation. So that sample means we have a finite set of individual cells. They undergo DNA sequencing, and then the data roughly looks like this. So it's a binary matrix where the rows are indexed by mutations and the columns are indexed by cells. And we're just indicating for each mutation in each cell whether that mutation is present in the cell or not. There can be missing data, of course, and there can also be false positives or false negatives indicated in red here. So starting from that binary matrix, we would like to reconstruct the tumor phylogeny. So that's a phylogenetic tree, such that the individual cells label or form the leaves of the tree, and the mutations occur on the branches of the tree, such that, you know, if you trace out a path from the root to the leaf, the cell that you reach is defined genetically exactly by the mutations on that unique path to that particular cell. So this object here we would like to reconstruct from the observed mutation data in individual cells. Now, the phylogenetic tree or cell lineage tree, as shown here on the right-hand side again, is sometimes not the most convenient way to do it, and we will often use the so-called mutation tree, shown on the left-hand side here. In the mutation tree, the vertices are actually mutations rather than cells, and the cells are then attached to the mutation tree. So here are the mutations or even sets of mutations that label the vertices, and individual cells are attached to the mutation tree. And in fact, we will most of the time not care about the individual attachments of the cells, and we will only care about the mutation tree. That means the order in which mutations occur and the branching pattern of the tree, so the partial order of these mutations. And this is because we typically don't have any additional information about these individual cells. So in a sense, we're not really interested in relationships among cells, but more on the order in which mutations occur and the branching patterns. Now, these two representations are equivalent under some assumptions, in particular the infinite science assumption, and we will in the beginning make that assumption, but relax it later on. So let me start then by talking about how to reconstruct such a tumor phylogeny, and I will also come back quickly to the problem of mutation calling. So assuming you have M cells and N mutations, the tree topology is noted by T, and sigma is a vector that tells us for each cell where it is attached to the mutation tree, as shown here on the right-hand side again. Now, the basic model that's shown here is in a way the simplest model one can use. It has only two additional parameters, namely the false positive rate and the false negative rate of the observed binary matrix of the observed data. And the likelihood then, well, is the probability of that matrix, and it just factors over both the rows and the columns of the matrix, so over both mutations and cells. And for each entry of the matrix, we have the probability of that entry, and that's just defined by a confusion matrix that takes the expected value defined by the tree into account and then compares it to the actually observed one. You can have false positives and false negatives. So that's the simplest model you can use in the sense that it doesn't have branch length. It only has the tree topology. It does not care about the attachment of the individual cells, but it has two parameters accounting for noise in the data, because noise is a very strong factor in single cell sequencing data, and we definitely need to take it into account. And we do need two different parameters for false positives versus negatives, because these two rates are typically very different, with false negative rate being very high usually due to failure to amplify the alleles, or maybe one of the alleles, such that we have very, very high allelic dropout rates, which leads to a high false positive rate. Now, as I said, we typically don't care about the attachment of the individual cells. So that means we have to marginalize out. We're computing a marginal likelihood, and we have to sum here over all possible attachment combinations of cells. So this sum is in the first place over all vector sigma that account for all possible ways to attach the individual cells to the mutation tree. But it turns out that you can actually factor this over the cells. You can regard each cell individually. And then the computation time to compute this marginal likelihood is just linear on the number of cells, and also linear on the number of mutations, rather than exponential as it would be in the naive formulation of this probability. So that leaves us with searching the space of mutation trees. So these are labeled trees on n vertices, and there are n+1 to the power of n-1 such trees. That's, of course, still a large space, even for a moderate number of mutations. But at least the size of this space is now independent of the number of cells that we're analyzing. We can compute the likelihood in time linear on the number of cells, and the space that we are searching is independent of the number of cells. So in other words, this is a good setup for a moderate number of mutations and a large number of cells. And we're searching this space by doing Markov chain Monte Carlo, so Metropolis Hastings type of sampling in tree space, which has, among others, the prune and reattach step and a few other operations. Okay, I want to come back now to the original data. I told you this is a binary matrix, but of course, in reality, originally, it is the read data that we're observing from the sequencing experiment. And so I want to step back now and talk about the mutation calling problem in the first place, which is in fact very challenging for single cells. The data now is not just a binary matrix, but is the coverage that we're observing at each position in each individual cell, and the number of reads that support a certain variant, a certain SNV, again, for each position we are considering and each cell that we have. Now, it's clear that if you have good mutation calls, that you're in a better position to reconstruct the tree correctly. But it's also quite intuitive that if you knew the tree already, you would be in a much better position to call the mutations properly. And we're trying to take advantage of that by doing joint inference over the mutation calls and the reconstruction of the tree. And so the model I'm going to show you a little bit more of is called psi-finite, because we will now drop the infinite sites assumption, and I will explain briefly how to do the joint inference. So first of all, how do we relax the infinite sites assumption? We are still not considering any possible genomic event, but we are considering a number of additional events in addition to just a plain heterozygous mutation. So that heterozygous mutation is illustrated here in node 5. It occurs on one of the alleles. We are also considering now hemizygous mutations, so mutations that occur only on one allele after the other one has been lost. We are also considering now parallel mutations, so the same mutation can occur independently in 2 lineages of the tree within the same tumor. And we're considering now events that cause a loss of heterozygosity, namely either using the wild-type allele, as in node 4 here, or losing a mutant allele, as illustrated in node 6 here. With these genomic events, we are, of course, no longer respecting the infinite sites assumption, so we're generalizing now to a finite site model. Now, the probability of the data, which is the raw read, the aligned mapped read data now, looks similar as before. The big sum over tau here now, tau is the vector that accounts for the placement of mutations on the edges of the tree. So it plays a similar role as sigma before. We've been attaching cells earlier to the tree. Now we're attaching mutations to the tree. And again, you have to sum over all these possibilities. And again, it factors now over the mutations. So let's look at the contribution that one mutation gives now to this likelihood. This is now modeled as a mixture model. And that mixture model actually accounts for the different possibilities that are illustrated in the figure above. So it includes the possibility of a heterozygous mutation, a hemizygous, losing the wild-type allele, or the mutated allele, or a parallel mutation. And the point is that these individual contributions to the likelihood can all be computed efficiently, again, in time that's linear on the number of cells. And this is simply achieved by traversing the tree in a clever way. I mean, it's essentially a depth-first search of the tree. And, you know, you have to take partial sums in a clever way, and that allows you to compute this likelihood again in time, linear on the number of cells. Now, we are again doing a full posterior inference, or sampling from the posterior. And since we're modeling now different types of mutations, we can actually obtain the posterior distribution over these different types of mutations. So this is the posterior for a particular data set over a range of cells and mutations of a heterozygous mutation, then of a hemizygous, then of loss and parallel mutation. Now, especially with introducing loss of heterozygosity and parallel mutations, you're opening up a lot of possibilities in the tree, and it becomes easily unidentifiable and easy to overfit. So the model does try to handle that by penalizing mutation loss and parallel mutation. There's a parameter psi here accounting for that. And that parameter in the limit, as it goes to infinity, will again enforce the infinite sites assumption, so not allow neither loss nor parallel mutation. But when it is 0, it allows it in the full generality. So you want to pick this parameter somewhere in between, such that you are allowing violations, but you do not give the model too much freedom here. Just very briefly looking at the performance of this method, the first row here is about mutation calling. So this is looking at the ability to get mutation calls correct. And it's comparing the model psi-finite here with a variation of it that, rather than the full MCMC, includes just a more heuristic search, a hill climbing strategy to get faster to a result. It compares it with the previous version of the model that did not allow violations of the infinite sites assumption, and it compares it to MONOVAR, which is a single cell mutation caller, but not taking the tree structure at all into account. And you see immediately that not taking the tree into account, you have a considerable disadvantage here in terms of F1 score. You get much better calls by taking the tree structure into account. And now if we look at increasing fraction of parallel mutations, the previous version, not allowing that, of course, lost precision, and we are recovering this pretty much in the new version of the model. And a similar effect here is for mutation losses. Similarly, the tree reconstruction performance works again very well, even when there are parallel mutations and when there are mutation losses. So next I want to talk about copy number variations in individual cells. As you know, cancers, most cancers actually have copy number variations, have genomic rearrangements that lead to copy number variations. And most of them actually have a lot of these. So the question is, can we also take that into account in this approach of analyzing single cell data? Well, the short answer is yes, but the more honest answer is, well, it can be done, but it is much more demanding. And the problem is that the space of possible genomic events and hence of trees that describe this process becomes much, much larger now. So the way this works now is that we start by binning the reads over the genome, and then we are calling potential breakpoints. They give rise to these genomic regions, denoted here R1, R2, R3, and so on. And the tree that we're trying to learn now is called here the event tree, where each genetic event is a copy number change. And copy number change means it affects a consecutive segment of regions of the genome by either amplifying or deleting that region. So for example, event 1 here would be an amplification of the segment that comprises region 1 and region 2, and so on. And in this manner, we try to explain the copy number profiles that we observe in individual cells or in clones defined by sets of individual cells, as indicated here. Again, we're trying to learn this tree jointly with what we call the copy number states. So these are the called copy number states that we try to infer at the same time that we try to reconstruct their dependencies in the event tree. So just to show you one example, these are data from breast cancer xenograft that comprises 260 cells. It's data from Sam Aparicio. And the raw data is shown on the top right here. This just shows the normalized read counts over the entire genome and over these 260 cells. And what we're learning now, again in a full Bayesian fashion, sampling from the posterior, is the tree depicted on the left-hand side here. I mean, the map estimate of the tree is just shown on the left-hand side here, and the copy number states as shown on the right-hand side in the middle. So one way to think of this is that the tree serves to denoise the raw data, and it gives rise to the predicted copy number states. And two individual cells are picked out. They're illustrated here with these two triangles. So two rows in this matrix correspond to two individual cells. And their copy number profiles are shown at the bottom here. The first sort of greenish one actually sits right here in the tree. And I hope you appreciate that this copy number profile, the raw data, is obviously very noisy. It comes from a single cell, even though the binning smoothes out a little bit. And doing these copy number calls in a single cell individually, separately, would be extremely challenging, more or less impossible at this resolution. But it is feasible here because we do it in the context of the tree. So we're sort of sharing information and we're gaining power by doing these calls simultaneously for all cells, taking their relationship into account. The next step is maybe obvious, but again, computationally quite challenging, namely to integrate the model that I just showed you for copy number variations with the model I showed you before that, which was restricted to SNVs, to single nucleotide variants. And in fact, there was a very brave student in the lab who did that. So the method he calls COMPASS. So this is joint SNV and CNV single cell tumor phylogeny reconstruction. And in fact, he created this model in the context of panel sequencing data, although in principle it can be done for any other type of approach, whole genome, exome, and so on. So now the event tree accounts not only for copy number changes, but also for SNVs, for point mutations. And in particular, we have now a notion of allele-specific mutations in that model. And just to show you what can be done now, well, the first thing is maybe obvious, let's say, maybe clear. So here's one such phylogeny for a particular AML sample, where most of the cells have gone through this particular history. I mean, that's what the model suggests, that's what we estimated. And there is a subclone here making up 5% of the cells, 120 cells in this example, on chromosome 17. And just when you compare that to bulk data here for a SNP array, then there's no way to detect such a low frequency clone. So obviously you get to see no low frequency events that in a bulk analysis would not have been possible. Maybe more interesting is the next example that illustrates the allele-specific mutations. Here's a particularly interesting example where the model predicts that there were 2 different point mutations on chromosome 17 in the gene coding for TP53, a well-known tumor suppressor gene, of course. And the model suggests is that from here, 2 different subclones have developed, both of which are defined by a loss of heterozygosity on chromosome 17, but they are defined by the 2 different point mutations that occurred on these individual haplotypes, on these individual alleles. So in other words, we get to see now in much higher resolution, or we can infer at least, the particular genetic events that give rise to the observed diversity in the sequencing data of a single tumor. So next, I would like to take now the perspective that we can reconstruct these trees for individual tumors, and I want to look now at a cohort of trees that we obtained from different patients and ask the question, well, what can we learn across different patients from these trees? Are there recurrent evolutionary patterns, and are there evolutionary trajectories that maybe at the end of the day we can even start predicting in a computational fashion? So the particular data I will show you now, this is a joint work with Koichi Takahashi from MD Anderson. He works on AML, and we analyzed here initially 75 AML patients, later I think around 120. A total of more than 500,000 cells have been analyzed. And we used here the Mission Bio tapestry platform. So that's, of course, single-cell sequencing technology that uses a panel initially only of 19 genes known to be involved in AML. Later, we used a slightly larger panel than these 19 genes. So a typical data set for one tumor looks like this. So this is the set of mutations that have been detected for this particular tumor, just 6 mutations. Remember, it's just 19 genes here. And this is the scale of the number of cells. These are already 1,000 cells. So this is more than 8,000 cells that what we're seeing here. And just to come back to that early point, in this particular situation, just 6 mutations, there's only 16,000 mutation trees here based on these 6 mutations. So that's easy. We could even enumerate in that particular case. Whereas if you talk about the cell lineage tree, there would be a very large number of 10 to the power of 30,000 and so on. So this is clearly a situation, small number of mutations, huge number of cells, where the mutation tree is much more convenient to work with. So to start with, for a few patients, we have 2 different time points. So here's such an example where one sample has been taken at baseline, and then a certain treatment has been applied to that particular patient, which was initially successful, but eventually led to a relapse. And at that point, a second sample has been taken. Now the reconstructed tree is shown at the bottom here, and it's actually plotted left to right. So the root here is on the left, and then time, goes left to right. And the clonal composition of that first time point is shown in this first column here of nodes of that tree, and the second time point in that second column here. And as you see immediately, there's in particular this clone here that appears to have driven the relapse of the tumor. It was in fact already present at low frequency, 1.7%, 122 cells at baseline, but then became dominant at relapse. And in this particular case, actually that data fits very nicely to phenotypic drug resistance data that tells us that the particular mutation that defined this subclone here actually had the highest level of resistance as compared to other mutants that have been present at baseline. This was the one that, using that phenotypic data at least, would have been the one you predict to probably outcompete the others under treatment. Now, looking across these trees, we recognized some recurring patterns, something that I call a convergent evolution. So here's such an example where a mutation in NRAS seems to have developed independently. Actually, it has developed at the same position of that of the amino acid sequence, but not exactly the same amino acid change. So most likely that is a similar effect, but of course we can't tell. Similarly, these are two interacting proteins, IDH1 and IDH2 here, that again have developed independently in two different branches of the same tumor. There's even repetition of certain mutational pathways. This one here, NPM affecting NPM1, IDH, and RAS. So the NPMM mutation occurred here in this tree, IDH1 and KRAS. If you follow this path, it's IDH2. And then again, it's two different NRAS mutations, this time in neighboring positions, and similarly a mutation in KRAS. So the same, or at least very similar mutational pathway is developing independently within the same tumor. So in order to find such patterns, we first devised two statistical tests. And the first one was meant to detect patterns of co-occurrence. So co-occurrence is indicated here. So here we're asking whether a certain gene pair occurs more often in linear stretches of the tree than you would expect by chance. And in the second test, we're asking for what we call clonal exclusivity. So these are mutations that occur independently in different branches of the tree. And I showed you examples of that just a minute ago. So here we're asking among all trees that have this particular pair of genes, or mutated genes, do we find them on different branches more often than we would expect by chance? Now, these tests can be worked out, the combinatorics can be worked out, and I'm showing you right away the result of this analysis, which actually combines the two tests. So on the y-axis here is the effect of the gene pair occurrence test, and on the x-axis, the gene pair placement tests. So on the top right here would be genes that we predict to be clonally co-occurring. So they like to occur on the same lineage, and significant was actually only one of these pairs, FLT3 and NPM1. And on the bottom left here are gene pairs that we predict to be clonally exclusive. So they like to be in the same tumor, but they clearly prefer to be on different lineages of that tumor. And this again comprises here the significant results, at least comprises 8 of these pairs. Now, this is sort of a testing framework to identify specific pairs. The next step we took here was to devise a full probabilistic model, generative probabilistic model that gives rise to these trees and at the same time captures what they have in common, including patterns of clonal exclusivity and of co-occurrence. So here's again our set of mutation trees that we observe. And the model that we're using is based on the so-called mutual hazard network. The model is called TreeMHN because our model, unlike the original version of the mutual hazard network that was developed by Rudi Schill, our model will generate trees rather than individual genotypes. Now, from this model, we can compute individual evolutionary trajectories, their probability, the expected waiting time of mutations to occur. And we can also start asking, well, if you observe a certain tree, what is the probability of any of the other mutations to occur next? So what's the next most likely mutation? So this model accounts for waiting times explicitly. And the main thing of the model is that the rate of these waiting times, denoted by lambda here, accounts for interactions among mutations, denoted by theta ij. And this parameter, whether it's positive or negative, will tell us whether there's a positive or negative interaction between two mutations. And that again is interpreted as preferring exclusivity versus co-occurrence. So here are already the results for now 123 AML patients. And blue, so this is the adjacency matrix of that mutual hazard network. And in blue is evidence for clonal co-occurrence, and in red for clonal exclusivity. So you see a lot of interactions among some of the mutations and much less so among a large set of other mutations, overall confirming the results from the testing procedure I showed you before. But of course, now we are in a different situation, we're in a different position, we have a full generative model. So for example, we can ask for mutational pathways. You can compute them directly from the parameters of the model, and the most likely ones are shown on the right-hand side here. with their probability and with the expected time for these mutations to occur. And this is compared here to the observed frequencies on the left-hand side of this figure. Now you want to ask whether these predictions are sort of meaningful. Is this better than guessing trajectories by chance? And I will not go through the details, but how much observed pathways, mutational pathways, are enriched in the predicted ones, the ranked predicted ones is shown here. And our model here, compared to some other alternatives, actually does that better than these models. So in terms of predicting the next possible mutation, there's two ways in which we have assessed this. The first one is sort of retrospectively looking at the trees that we already have and giving the model a subtree of that particular tree, and then asking whether the mutations that we did not show the model would be among the ones it predicts, or how high they are ranked among the ones it would predict. And the second validation is for a very small subset of patients where we actually have trees from 2 time points. Then, of course, we can ask whether the mutations that we observed in addition at the second time point are among the ones that we predict, or how highly they are ranked among the ones that we predict. Now, first looking at the retrospective assessment, this is shown here, and we're comparing the log odds ratio of our model compared to a baseline model that just takes the frequency, the marginal frequency of the mutations into account. And again, comparing it to a number of alternatives here, it turns out that our predictions are significantly better. So there is a certain, let's say, amount of variation in that sequencing data in the evolutionary process that is actually reproducible and can be predicted. And similarly, for the longitudinal data, again, this is only 8 or so samples that we have, and I'm showing you the percentile ranks here of the predicted mutations, and our model is here in the first column of the green part, showing consistently very high ranks, whereas other methods are sometimes better, most of the time worse, and overall cannot predict this as well as we can do here. Okay, the last, I don't know, 3-4 minutes I may still have, I want to talk about now analyzing single-cell DNA and RNA sequencing data at the same time. So now we're trying to integrate single-cell DNA and RNA data into one model. So the problem is now as follows. Again, we have a tumor evolving and sampled at a single point in time. Now we're doing single-cell DNA sequencing as before, and I will only refer to copy number trees in this particular example. But at the same time, from the same sample, we did single-cell RNA sequencing, which of course includes other cells that are part of the tumor, but we are particularly interested again in the tumor cells and in the variation that we observe among the transcriptomes of the individual tumor cells. There is, of course, quite an amount of transcriptomic diversity in these samples, and there's also genetic diversity, as we have seen. So the question is, how do these two relate to each other, and how can we analyze both of these jointly? Well, the model that Pedro Ferreira actually came up with is called SCATREX, and it takes as input the clonal CNV or CNA tree, so that's the tree that has as events only copy number alterations, and it takes the single-cell gene expression data. And what it tries to do now is to map the expression data onto the clonal copy number tree, indicated here by the additional gray node. So this is what we call the augmented tree. And if you do this, you are able to separate actually the effect that the copy number change has on the transcriptome versus other effects that the model estimates to be present in the data. Now, just to show you very briefly how this works, so we obtain this augmented tree via a so-called nested tree structured stick breaking process. So this is a generative stochastic process that will generate these trees, and it's nested in the sense that there's a tree for the copy numbers, and within each node of the tree there are subtrees that account for additional transcriptomic diversity of all the cells from that particular clone. So all of these cells in A here, for example, are genetically, regarding copy number, identical, but of course not necessarily identical regarding their transcriptomes. So we're trying to account for that diversity. And it's again modeled as a tree because some of these changes can be inherited, either because they have another genetic or epigenetic basis that we haven't observed in our data, or maybe through other mechanisms. So we count now for copy number events and for transcriptional change. Now, I will not go through the model in great detail, but I want to show you that the observed read count at the end of the day is modeled by accounting for 3 different types of factors. The first one in brown here is, of course, the copy number level, essentially assuming, that the amount of mRNA is proportional to the amount of DNA that you find at any single locus, any gene. The second factor here, what we call the cell state factors, they are independent of the copy number, orthogonal, but they are inherited along the tree. And finally, in the reddish color here are noise factors. So these are factors that are independent of the tree, and they account, well, for any type of noise, including, of course, biological factors such as cell cycle and so on. Well, what this model allows you to do, one such augmented tree is shown on the left-hand side here, well, is to get an overview of all the cell states, of course. You can compare CNV induced expression versus the observed expression. You can trace individual copy number changes along individual lineages of the tree and ask how heritable they are. And maybe the most straightforward is to do a gene-specific analysis and to compare the copy number tree with the observed cell states. And in other words, you can find now changes in gene expression that are not driven by the copy number change. And so they are more likely, again, to be either due to other genetic factors or triggered by the environment of these particular cells. And here's one such example for immune genes, HLA-A and HLA-C. The copy number tree is on the left-hand side with the estimated copy number states. And then if you look at the cell state factor on the right-hand side, you'll see that one of these estimated nodes, A0, so this is a sub node of clone A, actually has a negative factor here. So that means the level of expression is much lower here than we would expect based on the copy number state and, of course, also the other noise factors. And it's a similar situation for the other gene. And again, we're doing a full posterior inference here. So if you look at these posterior distributions, you see that for most of the nodes, it's around 0. So there's not much going on, except at A0 here, where the posterior puts its mass very far away from 0. So this is a clear indication that there's something going on in this particular subsets of cells that is not driven by the copy number change. Okay, with that, I would like to wrap up. So I hope I could convince you first that joint single-cell mutation calling and phylogenetic tree inference can be done efficiently together, jointly, and that this improves not only the mutation calls actually, but also the quality of the tree that you obtain. Second, we try to generalize that, first of all, to copy number changes. That turns out to be more challenging, but again, will improve inference of copy number states and copy number profiles. We can integrate these 2 aspects, SNVs and CNAs. I showed you how to integrate other multi-omics data, well, more specifically only transcriptome data, but we're also working on mapping other types of single-cell data onto these trees. So we think of the tumor phylogeny as the backbone that serves, or can serve at least, for integrating multi-omics tumor data. I explored with you a little bit finding recurrent patterns of tumor evolution, in particular of clonal exclusivity and clonal co-recurrence. And the model I showed you actually allows us to some extent to predict tumor evolution. And I think that will have a huge impact in the future when it comes to designing treatment strategies from an evolutionary perspective. So with that, I would like to stop here. I mentioned, or at least showed you most of the people who have been doing the work. I want to mention in particular Pedro again, Jack Kuipers and Shang Luo, and also a few former alumni. Etienne worked on the joint model, Jochen on sci-finite, Katharina, and Ariane. Thank you for listening, and I'm happy to take questions.
Thank you very much for this great talk. We have around 10 minutes for questions. So live attendees who would like to ask questions, please use the microphone that is here in the center of the room. And online audience, use the Q&A tab on the right of your window and enter your question. We'll read it aloud and answer it. Okay, so let's start with the online question. So there is an online attendee asking, have you considered the possibility that clonal exclusivity can be an indication of synthetic lethal relationship between two genes?
Yeah, absolutely. That's a very good question. I guess that I didn't say that, but that's the way we think of these patterns. We think that this is at least one reason they may arise through synthetic lethal interactions. The other, in a way, more modest or whatever interpretation is, that just together they do not confer any additional selective advantage. That's somehow a milder version of that, let's say. But absolutely, this is what we're trying to capture, and this is why we think these pairs are interesting. Because if these effects occur, one may also have therapeutic means to exploit that.
Thank you. Let's have a question from the live attendee.
Does it work now? So does your single cell mutation calling method rely on linkage of the SNP sites on the genome? I guess another way, does it work on whole exome sequencing data of single cell?
I'm afraid I couldn't hear it. Maybe Marinka can just repeat the question.
Yes, so the question was about, perhaps you can come up here and ask it.
I was just wondering, does your single cell mutation calling method rely on the linkage of the SNP data, or will it work on single cell whole exome sequencing?
I didn't get the last part, but maybe I start answering. So you're asking about linkage of mutations. So that is something we did not take into account. There are a few methods. I think there's something from Peter Park, and there's another method that try to do this. I think we feel, let's say, that there's not so much you can get out of it when it comes to short read sequencing data. Because on short reads, you don't observe a lot of linked mutations because they are short. So we didn't explore this any further, but others have done it. And there are probably situations where it is very beneficial. We felt for our application here, it is, let's say, not the most important thing to consider.
I see. So the second part was, in this way, do you have to use whole genome sequencing, or can you just apply this to whole exome sequencing? Would that work?
Yeah, absolutely. You can. I mean, I didn't probably say it very properly, but some of the data was panel sequencing data that we analyzed. Some of the data was exome, and some was actually whole genome. So in principle, you can, you can apply everything to every type of data, but of course it will not always be informative. So in particular, the whole genome data that we have, this is of course low pass sequencing data, so very low coverage, 0.1x or so. And at least in individual cells, you cannot really call mutations, of course. But after binning, if the bin size is large enough, you can talk about copy number variations. And after grouping cells together, so after identifying clones, you can even try to also call SNVs. So, depending, and on the other hand, if you go to panel sequencing data, depending on what that panel is, it may not be very informative about genome-wide copy number alterations, but it can, depending, on how amplicons are distributed over the genome.
Thank you very much. Niko, can you hear us? Bill Noble at the other microphone.
I can hear you, I think.
Okay, good. There's a really interesting talk, and I think you showed how various methods for analyzing the DNA sequencing data, and then you showed how to combine it with RNA-seq. I guess I'm wondering, how much mileage could one get if you only had the RNA-seq available? Can you make any of these kinds of inferences, even if they're somewhat imperfect, just from the RNA-seq data?
Yeah, that is a great question. And let me first of all say that this is obviously very relevant because there's much more RNA sequencing data out there, also including for tumors, obviously, than single-cell DNA sequencing data for different reasons, but that's what it is. So if we could do anything close to what we can do with DNA with RNA, it would be fantastic. Our attempts have been not so successful so far. I mean, we did try to do that. In particular, copy numbers you can try to call from RNA, but it is more noisy, and of course, it's only the expressed part of the genome that you get to see. So, the honest answer is that we have not been extremely successful in doing it. And there are clearly limitations. But I think it's worth further trying to explore it.